Mitochondria. Definition of mitochondria: these are membrane-bound organelles located in the cytoplasm of all eukaryotic cells. The number of mitochondria found in each cell varies widely, depending on the function of the cell it performs. For example, erythrocytes do not have mitochondria, while the liver and muscle cells have thousands of mitochondria. Structure of mitochondria: they are rod-shaped or oval or spherically shaped, with a size of 0.5 to 10 micrometers. Mitochondria have two special membranes, outer and inner membrane. They have a mitochondrial gel matrix in the central mass. The membranes bend into folds known as cristae. Functions of mitochondria: their primary function is to generate energy for the cell, i.e. they are the power generators, producing energy in form of adenosine triphosphate, ATP, by converting nutrients and oxygen into energy, enabling the cell to perform its function and to also release excess energy from the cell. Mitochondria also store calcium, which assists in cell signaling activity, generating cellular and mechanical heat and mediating cellular growth and death. The outer membrane is permeable, allowing the transport of small molecules and a special channel to transport large molecules. The inner mitochondrial membrane is less permeable, thus allowing very small molecules into the mitochondrial gel matrix in the central mass. The gel matrix is composed of the mitochondria DNA and enzymes for the tricarboxylic acid (TCA) cycle or the Krebs cycle. The TCA cycle uses up the nutrients, converting them into byproducts that the mitochondria use for producing energy. These processes take place in the inner membrane because the membrane bends into folds called the cristae, where the protein components used for the main energy production. System cells, known as the electron transport chain, etc., is the main source of ATP production. In the body, the etc. involves several sequences of oxidation-reduction reactions to transport electrons from one protein component to another, thus producing energy that is used for phosphorylation of ADP, adenosine diphosphate, to ATP. This process is called the chemiosmotic coupling of oxidative phosphorylation. This mechanism gives energy to most cellular activities, including muscle movement, and they power up the general brain function. Some, if not all, proteins and molecules that make up the mitochondria come from the cell nucleus. The mitochondrial nucleus genome has 37 genes, of which 13 of these genes produce most of the components of the etc. However, mitochondrial DNA is very vulnerable to mutations because they don't possess a large DNA repair mechanism, a common element found in other nuclear DNAs. Moreover, reactive oxygen species, ROS, also called free radicals, are produced in the mitochondrion, because of the preference for abnormal production of free electrons. These electrons are neutralized by antioxidant proteins in the mitochondrion. However, some of the free radicals can damage mitochondrial DNA, mtDNA. Equally, consumption of alcohol can cause damage to the mtDNA. Because excess ethanol in the body causes saturation of the detoxifying enzymes, leading to the production and leakage of highly reactive electrons into the cytoplasmic membrane and into the mitochondrial matrix, combining with other cellular molecules, forming numerous radicals that significantly cause cell damage. Most organisms inherit the mtDNA from their mother. This is because the maternal egg donates most of the cytoplasm to the embryo, while the mitochondria inherited from the father's sperm is destroyed. This causes the origin of inherited and acquired mitochondrial diseases due to mutations transmitted into the embryo from the maternal and paternal DNA or maternal mtDNA. Such diseases include Alzheimer's disease and Parkinson's disease. When mutated mtDNA accumulates over time, has been linked to aging and the development of certain cancers and diseases.